Hey, good afternoon, guys. I've just set a new homework for you. I've just set a dry lab homework looking at protein structure. I just want to give a short video just to give you a little bit of help in getting it up and running. So a dry lab is different from the usual wet lab in that it's a kind of investigation similar to a laboratory, an investigation done on a computer instead of in a regular wet laboratory where you might do, for instance, organic synthesis or something like that. And we all know about the idea that the protein structure, so the shape of the structure and the polypeptide, is what causes it and leads to its function. And the shape is, of course, talking about the orientation of atoms in space. And so we want to kind of understand where they are in space so we can understand more about the structure, understand more about the function. And sometimes there can be a protein that we don't know what it does, and we can figure out what it does by its structure, or perhaps we don't know how it does it, and we can figure out from the structure. And that can be important in a lot of things like drug development and things like that. So to find the homework, just in the quizzes, then here it is as homework. You can click it, and there's just a short kind of description of what to do. I'd like you to complete it using the protocol attached, which is here. To write a short reflection, and by reflection, I do mean just literally just a reflection on how you feel about this lab. Did you find it interesting? Did you find it boring? Did you learn anything? What did you learn? Just maybe 5 to 10 sentences on what your experience was in doing this lab and what you took away from it. I'd also like you to post the results that we'll collect in there. So we're going to be a Word document. You'll write your reflection, post the results underneath. These are going to be screenshots. And then underneath that, just answer some questions, which I've highlighted there in red. And then upload the document by the deadline. If I bring up the helper text, the instructions, here they are. And like I said, the orientation of atoms in a protein is critical to understanding its structure and thus understanding its function. And you can find a lot of these structures already on the PDB website, which I've shown you guys. And the PDB website is great. It's great storage of structures that have been solved and uploaded by researchers. And you can explore them around and look around a little bit. But it's limited in its power. And so we can use this new program, UCSF Chimera, which is a visualization program to visualize these structures. And if we just click to go to the website, you can bring it up here. This is the Chimera website, and you'll need to download the proteins. And actually, by sheer coincidence, protein of the day is actually the protein I studied in my postdoc for years, this peroxyredoxin here. But this is the website, and we do not want Chimera X. We want to use original Chimera. You can click here to download, and then you can download it for Windows, Mac, Linux. Click to download, run the program, install the program onto your home computer. If you can't do that because you don't have the facility, we've got computers up here on top floor of the chemistry building, NSB, third floor, over by the faculty offices. There's computers. They have this software on it. And it may be in the computer labs as well downstairs. But download and run the software, and that should install it. So it should be here. Now you can open up the software, and this is the software. So we need to follow the instructions that are written here. But very briefly, we'll go to the PDB website, open it up. We want to search. And then we want to search by structure keywords. And we want to, so this is going to do an advanced search, and we want to search by keywords. And the keywords that we want to search are this one here. Paste that in, and it will search and find it. And what we've searched for there is this term, structural genomics, unknown function. And it's saying, well, the structural genomics bit is not really so interesting. It's the unknown function. So what we're searching for here are proteins that we don't know what they do. These are proteins which the structure of them has been solved, but we don't know what it does. And so in this first lab, I'd like you guys just to familiarize yourself with the software, but in the later lab, you're actually going to do some research yourself and actually try to figure out what the function is. But what we have here is just a bunch of proteins that we don't know what they do. And we can refine the search. For instance, we can look just by human or by date in which it was discovered, family of what they think it might come from. But notice that there are 135 structures. Only one of them, they're like, well, it's a transferase. Don't really know much else about it, but we just know it's a transferase. So there's a whole bunch of different things we can use to narrow it down. We can just look at our protein structures. And a nice one, which I think works really well, is this one here, 1HTW, which I say to search for. And here it is. And if you, this is the structure of it, and there's some information about it. And if we click on structure, we can look at it in 3D. Here it is, the protein with ADP, magnesium. So we know that those are substrates, ligands, but we don't know what it does. If I click left mouse click, I can move it around. Or I can scroll in and out, or I can move it around with the right mouse click. But this is the structure. This here looks like the ADP rather, and this here, this is the magnesium. There's the ADP, there's the adenosine, and then phosphate 1, phosphate 2, that's a diphosphate. And this magnesium here is helping out. Again, it's a cofactor, inorganic ion. And what it's probably doing is that these phosphates have got negative charges to them. And magnesium's got a positive charge. So it's acting as a counter ion to help the ATP bind. But, and this tool is great. And you can see here, here's the helix. And here's a strand. There's some strands arranging together to form a sheet, and the turns between them. And here's the protein structure, the amino acid sequence. But the amino acid sequence is up here, but this only gives us kind of limited information. And we want to visualize it with a more powerful tool, and that's our Chimera. So what we can do is we can take this sequence here. You can download it if you like, download it to your computer and then open it, or you can just take the sequence 1HTW. We go to Chimera, file, and we can open up a saved file, or we can fetch by ID. And it was 1HTW. Click fetch, and boom, here it is. Here's our protein. Looks like it looks a little bit different here. The reason why it's different is because there are 3 of them. And I'm not sure exactly, maybe it's because it's a homo trimer, trimer 3, homo they're the same, or it might just be that they visualized it 3 times, I'm not sure. But this has got the protein, and we can again do the same thing. You can click and drag and move it around, zoom in, zoom out. A middle click now moves it around. You can visualize it in different ways. You can see there's the magnesium, and look, there's 1, 2, 3, 4, 5, 6 coordination bonds. One of them is with a negative charge of the phosphate. Looks like one of them is with a glutamic acid, and one of them with an aspartic acid, and then 3 of them are just with waters that are hanging around in there. But this is really this is the structure, and remember that it's showing here. This is the ribbon structure. That makes it easy for us to see identify the secondary structure, the helix and the strands. But that's not actually what it looks like. It's a cartoon representation over the top. What it actually looks like is if I go select chain, I want to select the alpha chain. I'm just going to hide this first of all. And if I was to hide the ribbon, so it doesn't show it anymore, and then show the atoms, this is what it actually looks like. if I select and clear the selection, get rid of it, this is what it actually looks like. And this looks to me like nonsense. I can't understand what's on there. That doesn't make any sense to me. But what I can see is all of the individual atoms that make up the structure. For instance, I can see right here, that is right here, this is a phenylalanine. This here is a glutamic acid. So what I should say is this beigey color is are carbons, the reds are oxygens, and the blues are nitrogens, and the yellows are sulfurs. And it looks like that's a tyrosine. This is a lysine. So I can see all of the individual amino acids. This is an arginine. So I can see all the individual amino acids, but I can't make sense of it. I can't see where the secondary structure is. It's just nonsense. So what I can do is I can select that chain A, so protein A, and I can hide the atoms and show the ribbon. And it's a cartoon that isn't actually what the thing looks like, but it helps me as a visual tool as a human being to understand what's going on here. So what I'd like you guys to do first of all is to there's 3 images here, and we only want one of them. So follow the instructions where you want to climatize yourself the software. You see it's a homotrimer. So we want to delete chain a B and C, or a protein B and C. And so what we can do is we can select A, invert the selection. So invert, we've selected A, and then invert, so select everything else, and then delete. So select A, select, invert, action, atoms, delete. Now we've got just one. And now we want to play around with the representation. So I want to take images of the protein in the full atomic. Actually, don't worry about the surface and ribbon. Just take the image in the full atomic. And so we want to go for select. Select residue. Okay, yeah. Select residue, so amino acids. Select the standard amino acids, and then it wants to show these. And then if we go to ribbon, hide, and now we have the full, and then select clear selection, and this is the full atomic view, that gobbledygook. But if we click File, Save Image, and I'd like you to save it as an image. File, Save Image with this size and transparent, and that will, and then save that, and that's going to go into your report underneath the reflection. Remember, it's reflection, pictures, results, and then the questions. So that's the first result. It's going to be this picture. Now I want to go back, actually. So select and hide the atoms and show the ribbon, and go back. So reverse, say here, reverse the previous steps to go back to the ribbon view. And now we want to, if we go to, we'll clear the selection, tools, sequence, sequence, and this is now showing the sequence of the protein structure. So this is the amino acid sequence of that protein. And we can see that some of them are highlighted in blue, green, and some are highlighted in yellow. And if I just click and swipe and drag and highlight this first bit here, what has been highlighted? What have I exclusively highlighted there? Or even if I click and select this one, what have I highlighted here? What have I exclusively highlighted? What's been highlighted there? I'll do it one more time. Or even just the bit that's not green or yellow. If I highlight this, what have I highlighted there? Think about that. Actually, I think, yeah, if I click this, this is highlighting all of the greens. What have I highlighted? What is it exclusively highlighted in green there? If I click the yellow, it's going to highlight all of those. What have I now highlighted here? So This is a tool to highlight certain secondary structures. And what the instructions tell you to do is to so what it is that these are helixes and these are strands. And notice interestingly as well in the sequence it goes strand, helix, strand, helix, strand, strand. Even though the strands are next to each other in 3D shape, they're not next to each other in sequence. Strand, helix, but the strands are next to each other. That's kind of interesting. Once the instructions say to select the first helix and then show the atoms. So first helix, swipe and select the first helix. There it is. Then click Actions, Atoms, Show. Now it's the first question. Can you identify the amino acids in the helix? What do you notice about the amino acids which are facing outwards versus facing inwards? So we look at the protein. Looks like these ones are facing inwards, right? And these ones are facing outwards. What do you notice about the inward ones? What type of proteins are the inward ones? What type of proteins are the outward ones? That's the question I'd like you guys to answer. That's highlighted here in yellow and red. And then finally, what I'd like you to do is some analysis of this helix here. If we click, and we're going to do the analysis, we're going to look for the hydrogen bonds and say, hey, can you show us where there are hydrogen bonds? We click Tool, and then it is Structure Analysis, Find Hydrogen Bonds. And you need to tick some of these. I want to tick color not finding precisely, and then with both ends restrict to find only with both ends selected, I believe, and then click OK. And now it has shown hydrogen bonds which it thinks might happen in orange, and hydrogen bonds which it's confident will happen in blue. And what do you notice about the blue ones now? This is what I was talking about when I was talking about protein structure. See, they are every 4 amino acids, they're lining up. Every 4 amino acids, they're lining up in order to stabilize in the helical formation. And even every 4 amino acids, you may get side chains between this lysine and this glutamic acid. You can see that hydrogen bond is formed between them. Or maybe it looks like hydrogen bond between this, what is this? Glutamic acid again and serine. Or aspartic acid might be. Yeah, see. There's 1 carbon, whereas this one's got 2 carbons. But you get the idea. So hydrogen bonds are forming every 4 amino acids with the backbone, and also with the R groups as well. So, and then it wants to save an image of this. So again, same thing. File. Oh, let's select. Click Selection, File, Save Image, and save this image. And then that's going to be the second image. And now you're going to have your first image with the full atomic, your second image, which is showing the hydrogen bonds, and then your answer the questions. So you're going to generate a post-lab report, write a short report reflecting on using the software, answer the question in point 10 here, and then paste the images from steps 8F and 12. And I've written here, take images of the full atomic surface and ribbon. I've only given instructions for the full atomic. I don't want you to do the full atomic. In fact, I might even delete this text and re-upload the document. I think I'm going to do that. But yeah, just do that and then post it onto Blackboard. I think that all in all, this will probably take you between 1 and 2 hours, so it shouldn't be too bad. Just follow the instructions. If you have any questions, send me an email. Thank you very much.